Welcome to Virology Live. This is session number 17. Today we are going to talk about persistent infections. Last time we talked about acute infections, and you remember an acute infection we defined as one that is rapid and self-limiting. And today we'll talk about those that are long-term infections and, in fact, last the life of the host. These persistent infections are stable. They are characteristic for each virus. And today we'll go through a number of different examples of persistent infections so you can get a sense of that. And I would say that in most cases, persistent infections actually start as an acute infection that is simply never resolved. So here is our graph of general patterns of infection that we saw last time with our acute infections on the top here. We're looking again at virus production on the y-axis and time on the x-axis, with the blue line indicating virus production and the red bars periods of disease. And so an acute infection, rapid and self-limiting, peak of virus production may or may not be accompanied by disease. And below them are different kinds of persistent infections. And all of these are persistent infections. They have different names, but I don't think that really matters. Here, the number 2 is a latent infection. Again, it's a persistent infection, but it has the name latent because the genome is silenced, at least for herpes simplex virus types 1 and 2 at different times. So you have an initial infection where you have virus production and disease. Then the infectious virus disappears, and then at subsequent times it will reappear again either with or without disease. So these inter-virus production periods, the genome is called latent because it's there, but it's silent. And we'll talk about that today. Then we have here in the third panel a persistent infection that is asymptomatic. So there is an initial acute infection without any disease. Virus goes to a high level, and then it remains at high levels throughout the life of the animal in the absence of disease. And an example of that would be lymphocytic chorial meningitis virus in mice. At least when inoculated intraperitoneally, if you put it right in the brain, you will kill the mice. And in humans, JC virus. And as we'll see today, polyoma viruses cause many persistent infections where the virus is reproduced continuously in the absence of symptoms. And then the last panel is, this is HIV/AIDS. I'm not sure why it's called persistent pathogenic. Frankly, this is from Principles of Virology, and this has been in it for the life of the textbook, 5th edition now. But this is just another form of a persistent infection. You have an initial acute infection that's not cleared. You have disease, and then the virus goes to a low level for many, many years, low virus production, which you can detect, and then at the end, it could be several years or more, up to 10 years. The virus reproduction then increases and the host dies, typically of opportunistic infections that can't be cleared because the virus is destroying the immune system. So we have HIV type 1 doing that, and we'll talk about that in a separate lecture. Human T lymphotropic virus has this pattern, and measles virus-associated SSPE, which we'll talk about today. And so today's cast of characters are shown here. I think it would be nice if you could look at all of these and figure out what they are by their icons. These persistent infections that we'll talk about today and all others, they occur when the primary infection is not cleared by the immune response. And depending on the virus particles, proteins, and genomes continue to be produced, you'll see there are differences. And viral genomes remain after proteins are not detected. That's how you get a persistent infection. The virus genome needs to be there. And paramyxoviruses, polyoma viruses, Hepatitis B virus, Hepatitis C virus, and herpes viruses are the icons. There's no single mechanism for persistence, although, say, herpes simplex 1 and 2 have a common mechanism, but they differ from other herpes viruses like cytomegalovirus. But in general, if cytopathic effects are absent and host defenses are reduced, this is likely to lead to a persistent infection. Now, why is that? What does cytopathic effects have to do with it? Well, you should remember that viruses that don't kill cells don't activate innate responses as well. They don't cause as much inflammation, and therefore the adaptive response isn't as good. So non-cytopathic viruses, reduced adaptive responses, more likely persistence. And a key part of persistence is viral immune mechanisms. Viral immune modulation. Here's a table of persistent human viral infections. We will talk about most of these today. The ones that have an asterisk, the red asterisk, we're going to talk about those. And you can see here's a list of the virus, the sites of persistence in the body, and the consequences of kind of disease. Now, at the very top is adenovirus, which can persist in a number of organs. You can see mainly lymphoid organs. And we have no idea what the consequence of that is. I think it also, I know people who have recurring eye infections, conjunctivitis caused by adenoviruses, which is latent in local lymph nodes and periodically gets reactivated and causes eye infections. But we have our herpes viruses here, our hepatitis B and C, which we'll talk about. HIV, as I said, gets its own lecture. Polyoma virus, Measles virus, and varicella zoster virus. And this will tell you each of the consequences that arise when these viruses reactivate. For example, with the herpes simplex virus types 1 and 2, the reactivation may lead to a cold sore or genital herpes. A key mechanism of evasion is at the level of the cytotoxic T cell response. So cytotoxic T cells are specific, can be specific for virus-infected cells. They are, of course, activated during the initial immune response in the lymph nodes, as we talked about some time ago. And cytotoxic T cells that are virus-specific emerge. And these cytotoxic T cells bear T cell receptors that can recognize a single peptide produced from a virus. That peptide is typically displayed on the surface of a virus-infected cell in the context of the major histocompatibility molecule. So the orange peptide is displayed by MHC, and we've talked about how it gets there previously. And then the T cell receptor recognizes it. This recognition causes the T cell to release the effectors of killing, perforin and granzyme B, which cause the virus-infected cell to undergo apoptosis. This is a extremely important defense against disease, one that is important for many viruses, including acute viruses, including SARS-CoV-2, but it is largely ignored in the current narrative, which doesn't make sense to me. But the T-cell response is why you are continued to be protected against severe disease and hospitalization. At least that is my understanding. I received an email this morning from an extremely well-known immunologist chastising me for speculating on that, but we'll save that for another discussion. Now, this MHC1 system, so MHC1, major histocompatibility molecules or complex, that's what the C stands for. One, there's two of them, 1 and 2. MHC1 is on all cells. 2 is just on APCs, antigen-presenting cells. So here's a virus-infected cell and MHC. Inside the virus-infected cell, the viral proteins are being made, and those are processed by the proteasome and loaded through the transporter into the ER. They're then loaded onto MHC, which then goes to the surface where it can be recognized by the T-cell receptor. And at every step of this process, there is modulation by viral proteins. And as you can see in this slide, There's these red boxes mean inhibition of that particular step. So we have, for example, inhibition of the transport of the peptides through the proteasome by viral proteins. Actually, this one, the Epstein-Barr Virus, inhibits the proteasome. These herpes simplex and cytomegalovirus proteins inhibit transport of the peptides and so forth. But look at what we have here. Epstein-Barr virus, herpes simplex, cytomegalovirus, adenovirus, HIV. And these, by the way, the cytomegalovirus and HIV proteins pull the MHC from the surface and degrade it. So the cell is never displaying a viral peptide, so it's never recognized. It's blind to the T cells, the virus-specific T cells. These are all large DNA viruses, and they cause persistent infections. And this is part of the reason they can cause persistent infections, because they can prevent their genomes and infected cells from being eliminated. Even adenovirus down here is inhibiting the transcription of genes encoding MHC. Now, another mechanism of evasion that we have not yet talked about is the emergence of cytotoxic T lymphocyte escape mutants. And so what is this? So again, the cytotoxic T lymphocyte recognizes via the T cell receptor a peptide displayed by the virus-infected cell. And so what happens in viruses that establish persistent infections is that these epitopes, these T cell epitopes, may change to prevent CTL killing. That's what we mean by a CTL escape mutant. And we see this often with herpes simplex virus and hepatitis C virus and other viruses that cause long-term infections. And so what's shown here is MHC, this dark gray protein is MHC. And here is a peptide fitting into the groove in the MHC. This is in a vesicle now. It's going to be sent to the surface where it can display the peptide. But if the amino acid sequence changes, which is shown by these blue amino acids to the left and right, if the amino acid sequence changes, the peptide will no longer fit into the MHC molecule. Or if it does, it's not recognized by the CTL. So here on the bottom is now the MHC on the surface of the antigen-presenting cell. And here we have an altered T cell peptide, which can no longer be recognized by the T cell. So normally the T cell receptor would recognize the peptide in the context of MHC, in this case 2, because it's an antigen-presenting cell. And then the T cell, the CTL, will kill the infected cell. But if there's an amino acid change, it may not be recognized by the T cell. So that's a CTL escape mutant. So that's one mechanism where the peptide changes, and you only need one amino acid, and the T cell receptor can no longer recognize it. The other is that there can be changes in the protein that prevent it from being proteolytically processed. So a peptide is never generated, and so it can't be recognized. So this is an important mechanism for persistence. Now, I have to tell you that we do not see changes in T cell epitopes, that's what we're talking about here, for the most part in any of the SARS-CoV-2 variants that we have seen so far. And you can go look up Alessandro Sette and Shane Crotty's work on this. They are the ones who are studying the T cell epitopes. They don't change in variants of concern. And I'm sure they're not going to change in the latest variants. And that's why you're protected against severe disease and death. Now, why don't they change? That's an interesting question. They do probably change, but it doesn't matter because, first of all, the infection is quick. It's a couple of weeks and it's over. Whereas in a persistent infection, it would make more of an impact. And secondly, if it did change, and let's say you transmitted a virus to someone else with a change in a T cell epitope, it wouldn't matter because their MHC is different, and so it would have no impact on their infection. So the only CTL escapes that matter are the ones that are in you. And that's why it's only in a long-term persistent infection that it can make a difference. And that's why the variants of SARS-CoV-2 can still be recognized by CTLs because the T-cell epitopes aren't changing. A few of them are, but they don't make an impact because there are more. And there are many T-cell epitopes throughout the viral proteins. Of course, the ones in spike are the ones that most people focus on. B-cell epitopes can change, but not the T-cell. And this is why CTL escape mutants matter only for the person that they arise in, not for others, because of the extreme polymorphism of MHC. Now, sometimes, in some instances, sorry about that. I wanted to make this slide bigger. In some instances, when a CTL engages an infected cell, it's actually the CTL that dies instead of the virus-infected cell. So this is a form of a viral countermeasure or a viral defense. Now, normally, this is put in place to limit immunopathology, this kind of restricting of T-cell killing to limit immunopathology. So it's a built-in mechanism. But the way it's mediated is that the CTLs carry a membrane receptor called Fas on their surfaces. And this binds a membrane protein called Fas ligand. When Fas ligand binds Fas on their surfaces, the receptor trimerizes, you get activation and induction of apoptosis. All right, so Fas binding to Fas ligand, as well as TNF binding to different receptors, but for us, it's Fas ligand here because this is the receptor is on the surface of the CTL. And so HIV and cytomegalovirus actually induce Fas ligand on the infected cell surface. And so when the T cell comes in to the HIV or CMV infected cell, Fas ligand binds Fas, and boom, the CTL dies. How insidious is that, right? So this is again, I want to emphasize, this is a normal process in us to limit immunopathology by getting rid of CTLs when they're no longer needed. Now, it turns out that certain irreplaceable organs or tissues, like those in the eye, the testes, and other places, they remain free of potentially destructive T cells by maintaining a high concentration of Fas ligand on their cell surfaces. And so this is why, in part, you can get persistent infections of the eye and the testes and other locations because CTLs aren't working there. They're killed. There's high levels of Fas ligand in those tissues. So another reason for persistence is this idea that there is reduced immune surveillance in some tissues to prevent damage. So cells and organs differ in their degrees of immune defense. And as I've just said, there's certain tissues that are high in Fas ligand, like the eye. So the vitreous humor there inside. You can imagine if CTLs get in there, that would cause a lot of damage. So there's a lot of Fas ligand. So if CTLs do get in, they are not allowed to kill their target cells. But the CNS, the skin, and other places are devoid of other initiators and effectors of the immune response. And we think this is because they're easily damaged by fluid accumulation, by swelling, ionic imbalances, all of which result from inflammation. And so persistent infections of these tissues are common. And the skin, for example, is continuously flaking off, and that prevents infection, as we've told before. The antiviral response to the central nervous system is distinct in the brain. It favors not CTL-mediated killing, but non-cytolytic clearance by cytokine release. But nevertheless, persistent infections can occur when infected cells are not cleared. Now, for many years, we thought that Ebola virus was a typical acute infection. But then a paper was published a number of years after the 2015 outbreak. Here it is in New England Journal of Medicine. Persistence of Ebola virus in ocular fluid during convalescence. And this was one of the first reports of a patient who had recovered from Ebola virus disease, EVD. And this was a physician who had worked in Africa, recovered, came back to the U.S. to work, and then developed unilateral uveitis, inflammation of the eye, of one eye, 14 weeks after onset of Ebola virus disease and 9 weeks after clearance of viremia. So historically, we have said, okay, when you have no more Ebola virus detectable in your blood, you're cleared, you're done, you can go back to whatever you were doing. And so this alerted us to the fact that the eye can apparently harbor infectious Ebola virus for a long period of time. And I think one of the reasons is that perhaps the virus gets in there, right? And then it is not cleared because high fast ligand in the eye. And apparently it can stay there in an infectious form. That's astounding to me that it can stay for years. And we also know that it also persists in the testes as well. In fact, there's been a recent flare-up of Ebola virus disease in Guinea, originating from a survivor of the original outbreak in 2015 who contained virus in seminal fluid for more than 500 days. So is this now telling us that Ebola virus is a persistent infection? It can be. These incidences are quite rare of these persistence in the eye and in the testes. But they do occur. So as with some of the other infections that we're going to talk about today, it can be persistent under certain conditions. And there's no reason why it has to be one or the other, right? In most cases, Ebola virus infection is an acute disease. It's over in a period of time and resolved. But in some cases, it can be persistent, and that's fine. We have to be okay with that. Now, in the this Guinea outbreak in 2021, these are some of the genomes that have been sequenced. And so what we have here is a phylogenetic reconstruction of the data from 300 isolates from the 2015 outbreak and 4 2021 genomes. And so what we have here is this dark gray box is the new 2021 lineage. So these are all of these genomes are the 2015 genomes right here. And you can see that we have 4 of them here from the 2021 Guinea outbreak, which is a new lineage. And you can see that these are essentially derived from one of these tips on the rooted tree, or on the phylogenetic tree. And so this is part of the information that leads us to believe that this virus persisted in a single individual for over 500 days in the testes, and this appears to have been sexually transmitted to start this new outbreak. So quite an interesting situation there. All right, so we're looking at mechanisms of persistence, and here's another one. Some viruses can actually infect immune cells, and we've talked a bit about this for a variety of viruses. Measles virus infection of antigen-presenting cells, for example. HIV-1 infection of Cd4 T cells, monocytes, macrophages, dendritic cells. This, of course, has severe consequences, as you can just guess without even knowing anything further. We've talked about how measles virus skews the Th response, the T helper response, how it can infect, and doing that by infecting APCs, but it can also infect memory B cells. So this can have an impact on persistence for sure. All right, let's take a quiz. Now, I'm going to stay here for this part. Which of the following are features of persistent infections? They last the lifetime of the host. Viral immune modulation is involved. Immune cells may be infected. They may occur in areas of reduced immune surveillance, and all of the above. And while you're looking at that, we can take some questions here. I notice, when the news cycle explodes, then we get a lot of downloads. I use Daniel Griffin's updates as a metric. So they post at midnight on Saturday morning here in the East. And then, at 6 a.m. when I'm making coffee, I check. And typically there's, 1,500, 2,000 downloads by then. But this Saturday it was 4,000 already. So I know, okay, people are freaking out about this news, so they go see what's happening here. Okay, let's get down here for our questions. Oh, yeah, good morning, Vanity. Thank you for moderating. And also, of course, Steph is here as well. And our Mr. Ozzy Cam, thank you all for moderating. I really appreciate the work you do. It's necessary. Okay, where's some bio questions here? Are these human infections always persistent in everyone, or some people manage to clear them? Some people can clear them, right? Like any acute infection, some people clear them. Yeah, not everyone. Although you have to understand that, the herpes viruses, almost everybody is infected with them. We'll see a graph of that at the end. So I think in most cases, maybe you get a second chance. Maybe the first one doesn't take. I think that's possible, but I don't see why not. But, it's very hard to know that because with time, the seroprevalence increases. Okay, my immunology is terrible. B cells interact with antigen. But we did mention that in one of the lectures. But I understand you're probably pipetting. And Hotel Papa says, is that responsible for eyes and testes in Ebola? And yes, of course, we went through that. Is it possible to be infected with several strains of the same? Well, it depends what you mean by strain, okay? If it is a serotype, then yes. So there are 3 serotypes of poliovirus. You get infected with one, you recover. You could still be infected with types 2 or 3. So serotype is the key there, because if the virus is the same, so there is one serotype of measles virus. And once you're infected, you're protected because there are no other serotypes circulating. Why do the eye and the testes have so much? Because it's a closed space, right? The eye is closed, the testes, and if you get inflammation, accumulation of fluid, they're going to swell, and there's not much room to grow, the eye or the testes, the brain as well, and that would not be good. That would cause damage. So that's the idea. What is Fas ligand? Fas ligand is the protein that binds Fas receptor to turn on apoptosis. It is normally there to get rid of cytotoxic T lymphocytes when we don't need them anymore. Okay? Which cells does Ebola virus need to get into to reproduce? Well, this is an interesting question. We will talk about this in a couple of weeks, I think, but it infects many cell types, essentially. And it gets into the blood, so it has easy access to the eye as well as other tissues. Can any virus become persistent if it happens to infect the eye? It could, although some could be. The CTL clearance is an important one, but there are other mechanisms, and it depends on the virus whether they would work or not. But I also think that the virus would have to remain infectious in the eye, and I think that's a high barrier to get over. And for some reason, Ebola virus can do that, which I would never have believed if you had told me that an RNA virus can last years, 5 years in the eye. I would have said no, but it's true. So I think it depends on the virus also. But yeah, it could in theory, depending on whether, if the infectivity doesn't last, it's not going to become persistent, right? So those are important issues as well. Does that explain herpes? No, herpes is a different story, which we'll talk about. But also, herpes can infect the cornea, and it's not persistent there. It's eventually cleared, but the clearance comes at a cost. I don't know how Ebola persists for so long, because those genomes are clearly derived from viruses that were circulating in 2015. If it had been reproducing in persons all the time, it would have been much more genetic change. How this happens, nobody knows. How it can retain infectivity. I mean, what we're saying is, in 5 years, it's reproduced a couple of times. And I find that remarkable. So we don't know. Nobody knows the answer to that. It is good that people come here when there is news. The problem is, about half of them are yelling at me for not covering this bloody variant. And I will cover it when I think it's appropriate. I'm not here to feed the news cycle. It never was the idea of TWIV. It's to explain science. And to the extent that we can, we will do current science, which obviously is important in a pandemic. Are there any persistent viruses which cause mild symptoms? Well, any of these, the Epstein-Barr virus, right, can cause fatigue. But any of these could cause mild symptoms as opposed to being symptomatic. Thank you, Lynn, for your contribution. Yes, it is too late. So the U.S. closing people from Africa, it's already too late. It's ridiculous. It's just a show to say, yeah, we're doing something. Can stress reactivate? Yes, we'll see that in a moment. Are Cd4 cells the same? No, they're different cells entirely. Cd4 cells do very different. They secrete cytokines, as we discussed, and NK cells kill cells. So very different functions and very different cells entirely. Yeah. There's no mechanism like cross-protection and superinfection. Yes, there is. There is superinfection exclusion for sure. We don't really talk about it in this course, but it does happen. EBV in the ocular fluid. I'm not aware of EBV getting into the ocular fluid and how it would and the consequences. I am just aware of herpes viruses and Ebola viruses. Is it possible to engineer fast ligand-free T cells? Ooh, that's interesting. So fast ligand receptor. Yes, fast receptor T cells. Fast ligand is the ligand. Yeah, well, but the problem is that you would not be able to control them then. They would not go away, and so you'd probably get a lot of immunopathology. So I'm saying that was not a good idea. Is there a relationship between persistent infection and the human virome? There might be. So in other words, could your virome somehow be associated with the outcome? Yeah, it could be, but nobody. It's been so infrequently studied, the human virome, that we don't know. But I think it's absolutely possible. Yeah. Herpes is around the lip. Why? We'll get to it. We'll get to it. I'm going to talk about that in a bit. Now let's get to the quiz. Now I have to go to here. All of the above, of course. Which of the following are features? They last the lifetime of the host. Immune modulations involved. Immune cells may be infected. They may occur in areas of reduced immune surveillance. It's all good. All true. All right, let's go through some examples of persistent infections. Measles virus. Oh, yeah, this was in our acute infection session last week, right? Yeah, it just illustrates that it's not black and white always. Here's measles virus, a paramyxovirus, extremely contagious. Look at this, 207,000 deaths globally in 2019, completely preventable by vaccination. It's just a shame, right? Lifetime immunity after infection, a classic acute infection. It comes and goes, you resolve, and you may get very sick, but for the most part it resolves. Or does it? Well, not always. About 1 in a million kids who recover from measles 6 to 8 years later develop SSPE, subacute sclerosing panencephalitis, which is an encephalitis. It's a brain infection, progressive, degenerative, 100% fatal. 1 in a million. So we still have SSPE because we have quite a few measles cases globally. Now, what's going on? This is very interesting, but we don't have an answer. You cannot find infectious virus in the brain. You can find nucleoprotein particles, which means the nucleoprotein bound to the viral RNA. You can find that in the brain, and apparently is able to spread between neurons via the synapse, right? From one cell to another via the synapse. The nucleoprotein, but there's no infectious virus. And so what's going on here is not known. We don't know if there are any changes in the virus that are important for this. All we know is that it's preventable by vaccination, but it is a consequence of a long-term persistence. And remember, measles virus infection is associated with encephalitis, about 1 in 1,000 cases. So the virus does get in the brain. It is neuroinvasive, and apparently it can remain in a form that is not infectious but able to cause disease. So that's an example of how an acute infection can become persistent. And it's rare, but it's clearly a thing. All right, polyoma viruses. I love this one. Here's a phylogenetic tree of polyoma viruses. We talked about these a lot in the first part of this course because SV40 is a model for understanding molecular biology, DNA replication, transcription, and so forth. But it turns out there are a lot of polyoma viruses. We have 3 genera here, shown by the blue lines, and the ones with the red asterisk. These have been found in humans. And so we have bird polyoma viruses, and we have Wookiee polyoma viruses, which originally isolated from a Wookiee, apparently. And these are all human viruses. The name actually comes from WU, Washington University, Karolinska Institute, Stockholm, Wookiee, okay? But, the polyoma virologists named this genus Wookiee. They could have named it something else, but it's pretty fun, I think. And then human polyoma, 6 and 7, less interesting, but those are the humans. And then there's orthopolyoma, which has bat and primate and so forth. But also we have BK, BK polyoma virus, which I always love to think of the hamburger one, but it's not. So there's nothing to do with BK. It's the initials of the patient from which it was isolated, as is JC. Not the person you're thinking of as John Cunningham. And then Merkel cell polyoma virus is a rare form of a skin cancer. Tropical spastic paresis polyoma virus. Right, so these are human viruses, and you can see some of them are associated with cancers and other diseases. But it turns out that most humans are seropositive for various polyoma viruses. You apparently get infected early in life through your family contacts, and you're infected for life. And these viruses, like JC and BK and other human polyoma viruses they're called that, they infect a variety of organs. They can infect kidney, intestine, respiratory tract, and many people excrete a lot of virus in urine, for example, 100,000 particles per mil in urine. So it's made in the kidney and it's being excreted. Now, there's some idea that there may be periods of more virus reproduction than other periods, but everyone's infected, more or less continually making virus, but apparently no damage to us. So all of you most likely are infected with polyoma virus. I certainly am. We don't know how they persist, but they're there. And I always tell people, look, the next time you go in a public bathroom, when you flush a toilet, you make an aerosol or a urinal. I think of it every time I go to a bathroom. And, some people before you, they don't flush. And then you go to flush it, you're probably inhaling their polyoma viruses. What are you going to do? There is one disease called PML, progressive multifocal leukoencephalopathy, which occurs in patients who are immunosuppressed. And in them, their polyoma viruses start to reproduce. And this one in particular, reproduces in the brain and causes a brain infection. And this one is associated with a treatment for multiple sclerosis. There's a monoclonal antibody called Tisabri, which is used to treat multiple sclerosis. It's an immunosuppressive antibody because many of the symptoms of MS are due to autoimmunity. But what this does, the immunosuppression causes the polyoma virus to start reproducing and cause brain disease. And so they have to take those individuals off the treatment. Anyway, I had a nice conversation a while ago with Bob Garcia, a polyoma virus out in Denver, Wookiee viruses. A lot of fun. All right, Hepatitis B virus. Remember Hepatitis B virus, the envelope virus with an icosahedral capsid? It has a DNA genome that's gapped and has a piece of RNA attached to it. It has a protein. This has reverse transcriptase in its reproduction cycle. Well, this is a human pathogen. It is transmitted from human to human by exposure to blood, which can happen during childbirth if the mother is infected. It can happen if you get a transfusion of contaminated blood during sex. Drug use, of course, tattooing. Can even get it in the hospital. And most kids nowadays should be immunized against Hep B because you never know when you're going to get infected, and it's just not a good infection to have. I had to be immunized because I work with human cells, which could be potentially infected with hepatitis B virus. Most adults, if you get infected as an adult, 95% of adults can resolve acute infection, but newborns cannot. Newborns have a problem. Only 5 to 10% of them can resolve an acute infection. And here are some numbers for hepatitis B in the world. 257 million estimated current cases. The Americas, about 7 million, but many, many more in the West. These are the WHO regions, right? The Western Pacific region, 115 million cases, 60 million in Africa. And Western Pacific includes all of these countries here. So there's a lot of viral hepatitis B in the world. Despite the fact that we do have a vaccine, we do have some antivirals, but they're not terribly effective. And part of the problem is that the antivirals do not get rid of the viral DNA, which remains in the nucleus of cells. The virus is not cytopathic, and so we can't get rid of that. And so I did a couple of TWIVs in Stockholm a couple of years ago, 2019. And there was an investigator at the Karolinska who was doing a strategy where instead of keeping patients on antiviral forever, you take them off, and then the virus begins to reproduce, and the T cells kill the infected cells. And then you put them back on. You do this on and off, and apparently this, it activates these DNAs in the cells, and you can kill the infected cells. So that's an interesting one to take a listen to. All right, so here's the pathogenesis of hepatitis B. Here we have on the left acute hepatitis B. So you should know what an acute infection is. So we're looking at, on the y-axis, different things. We have proteins, we have antigens, and we have antibodies. We have a liver enzyme here, ALT, alanine transaminase. And then we're looking at weeks post-exposure. So after your incubation period, you start to have a rise in hepatitis B surface antigen. It's a protein. We're looking at one of the viral proteins as a measure of infection. We are looking at DNA also here. You can see the green line. We're looking at antibodies. Here, first IgM and then IgG. And you can see there's a peak. It takes longer. It's not a weak infection. We're talking about months here, but there's a peak of viral antigen, viral DNA. There's liver enzyme in the blood, which is not supposed to be there. It indicates liver damage to liver cells. There are symptoms here, this little bar here. And then everything collapses, the infection is cleared, and you're left with antibodies. Okay, so that's an acute infection. As I said, 95% of adults do this. They can clear the infection. So it takes, 32, maybe, to get rid of the DNA, but, the antigen is gone in 6 months. So it's not a quick one. But there you go. And then we have chronic Hep B on the right here. And you can just see there's a difference, right? You have your initial onset after exposure, but then the liver damage continues. It doesn't go down to zero. The alanine transaminase, the viral DNA remains, the viral proteins remain, right? So this is a persistent infection, which happens in 5% of adults and most children who get infected less than a year of age. And interestingly, the symptoms mainly take place in this, what we call the acute phase. Of course, damage is done by the immune system, in particular cytotoxic T lymphocytes that kill the infected hepatocytes. But like all of us, cytotoxic T cells get tired. There is a thing called T cell exhaustion, where they just stop killing infected cells. And so when you have so many cells infected, that's what happens. And this is a problem with cancers too. And we can wake up those T cells by doing checkpoint therapy, right? And that's a key to reactivating or reanimating those T cells. And it may be a thing for hepatitis B as well. So you have T cell exhaustion, and so then the virus-infected cells continue to be infected, and then the T cells may reanimate again. They will kill some more cells, and then they get exhausted. So you have these cycles of killing and regeneration of the liver, and those cycles over and over, they cause liver fibrosis, damage to the liver tissue, and eventually can lead to cirrhosis and liver failure, and eventually cancer, hepatocellular carcinoma. That can happen after 20 to 30 years of chronic infection, and often asymptomatic. As I said, the symptomatic period, that acute phase, really that's all you would have there. And then you're infected for a long period of time. You're getting more and more liver damage, and you can get liver cancer. And one of the ideas is this continuous regeneration of cells. this is a recipe for cancer. We're going to see about this on Wednesday when we talk about transformation and oncogenesis. So the association of hepatitis B virus with cancer is one of the reasons why we want to get rid of it, because people will die of liver cancer. If we had a better antiviral or a better antiviral strategy, that would work as well. But certainly vaccination, but clearly vaccination is not reaching the people who need it. Common story, right? All right, so that is hepatitis B virus, and we have hepatitis C virus. It's a different virus entirely. This is a flavivirus. Can you name another flavivirus? I would hope so. Don't you dare say SARS-CoV-2. Another flavivirus would be West Nile virus or dengue virus, enveloped RNA viruses with plus-stranded RNA. And here, hepatitis C virus is different. It is not transmitted by mosquitoes. It is transmitted by exposure to contaminated blood, like hepatitis B virus, sex, drug use, tattooing, and during birth. We currently have 71 million people infected globally. And here's the map of that, which shows you the WHO regions, with most of the infected Europe, Eastern Mediterranean. It's kind of evenly distributed there. Western Pacific, Africa as well. Now you notice there's a lot of infection in Egypt, and there's a reason for that. I'm going to tell you what the reason is for that. So Egypt is the country with the highest prevalence of the disease with Hep C in the world. This increase started about 50 years ago when the Egyptian government was trying to eliminate schistosomiasis caused by a parasitic infection, right? So the Egyptian government conducted multiple mass treatment campaigns using an injectable drug to treat schistosomiasis, but they reused the needles without sterilizing them. So hepatitis C virus, which was not known at the time, but of course I told you is efficiently transmitted by blood, was inadvertently spread to many people, and by 2008, 1 in 10 Egyptians had hepatitis C. 1 in 10 as a consequence of reusing unsterilized needles. Now, when we talk about HIV/AIDS, just keep that in mind, how human interventions can help spread an infection. So it's the moral of the story. Use disposable needles or sterilize them. You know, there was a time in the world when we didn't have disposable syringes and needles, and we simply sterilized them. And that was in some places hard to do or inconvenient, so you get the consequence of that. All right, hepatitis C pathogenesis. Let's see here. Let's look at the bottom. We have a graph of what's going on. We have hepatitis C RNA on the left y-axis. We have alanine transaminase, the liver enzyme, on the right. And remember, we're measuring it in the blood, and it's not supposed to be high in the blood. It's supposed to be inside the liver cells, so its increase is a sign, and it's a sign, right? It's a sign because you have to measure it with a test. It's a sign of liver cells being damaged. And then months after infection. So we have exposure, and then we have a peak. So there are two outcomes here, clearance and persistence combined in one graph. So we have a peak of the red solid line, which is HCV RNA, right? And you can see that's going up and down here. And then the dashed line is the ALT, so that peaks and goes down as well. And, you know, by 3-4 months, can be cleared. But then in a number of individuals, the infection persists, and you can see here the RNA persists months, years, and so does, let's see, ALT, the blue dotted lines. It kind of drops a little, but there is some persistence over what the baseline should be. All right, so that's clearance versus persistence. So what do we have here? We have acute infection, as we've described here on this graph, and then after 3 to 6 months, 10 to 40% of the people will recover and no longer have any virus in them. However, 60 to 90% develop a persistent infection. And look, it's even called chronic here, same thing, persistent chronic infection. That can go on for 30 years. 2 to 30% of those people will end up with end-stage liver disease, cirrhosis, and hepatocellular carcinoma, very much like hepatitis B, resulting in death. And 70 to 98% will end up with an asymptomatic persistent infection. So some people, just like Hep B, the virus persists for years and apparently doesn't cause any issues. So this persistence is via multiple modulation mechanisms. That's at multiple levels, in particular at the level of interferon sensing and production. Now, for this reason, we developed many antivirals against Hep C, which we'll talk about in the session on antivirals. And this infection can essentially be eradicated. As far as we know, Hep C only exists in people, and a 12-week treatment of drugs can clear the infection. So it's theoretically eradicable. I don't know if it can be eradicated, though, without a vaccine, because new infections continue to occur because of drug use and so forth. It's hepatitis C virus. Now, part of the problem here is that opioid crisis in the U.S. has led to an increase in Hep C infections. So even though we know how to prevent it, people are into heroin, fentanyl, all these prescription opioids, and they have to be injected, and they're not using new disposable syringes. And so this is a crisis in the U.S. where we have a huge increase in Hep C, and also HIV infections as a consequence of the use of these drugs. So new infections and deaths are associated with these. Let me see if I have the numbers. The number of new infections has tripled since 2010, and 17,000 associated deaths. And also this goes for Hep B as well, not just Hep C and HIV. So that's why I say, even with an antiviral, you could take an antiviral and clear your infection, but if you're still taking drugs, you're going to get reinfected most likely. All right, it's time for a quiz. I have to go turn it on in Socrative, then I'll come back to the nice form. All right, which are shared features of persistent infections with polyoma virus, hepatitis B virus, and hepatitis C virus? This is a tricky question because you're going to have to think all three. A, genomes are present but not expressed. B, liver damage. C, kidney damage. D, virus particles are produced, or all of the above. What's in common? Okay, let's take some questions here. Yes, when viruses manage to downregulate MHC, the hour of NK cells come. They scan for proper density. Yes, and we talked about that briefly. So NK looks for absence of self, meaning absence of MHC. But you know what? Some of these viruses can counter NK by putting up fake MHC or doing other things to antagonize. And many of these persistent viruses that we talk about today can do that. Can I explain how epitopes recognized by T cells are different? Well, let's say you have a 1,200 amino acid spike protein. So here is a B cell epitope. It's a 12 amino acid sequence. And then you move a little bit down the protein. There's a T cell epitope. And then you move down. There's a B cell, a B cell, a B cell, then TTT. They're just different sequences, right, of protein. An epitope is a short part of a protein, 10, 12 amino acids. And so they're simply in different places in the protein. So if I drew a picture of spike, I could color B and T cell epitopes and they would be separate. Hope that helps. Oh, thank you, Rika, for watching every video. I'm glad you like how I explain things. It's in my head. But it took a long time for me to get to this point. Is SSPE a prion disease? I don't believe it is. There's no evidence that it is. But we will talk about prions towards the end of this course. Oh boy, they're so interesting. So interesting. Yes, so here's a great point from home. You mentioned SSPE incubating. I wish anti-vaxxers took that into account. I do too. However, they would tell you, it's 1 in a million. They're willing to take a chance with their kid. You know, you cannot reason. I should have said, with EVD persistent in the is there a pathway for full Ebola? That's a good question. I would say there should be, right? But I don't know of any case where in that person where the virus has been harboring that they developed full Ebola. Maybe because they've recovered and they have immunity of some kind, right? But I do know that it can be transmitted to if it's persistent in the testes, then it can be sexually transmitted, and that's been documented. But yeah, interestingly, those people don't develop Ebola as far as I know. Are those nucleoproteins found in brain remnants of past measles? Yes, actually. In people who don't develop SSPE, in a certain fraction of people, you do have viral genomes in the brain. Yeah, even with mumps. In fact, with mumps, I think over half of kids get mumps genomes in the brain, but mumps doesn't cause any neurological symptoms. Chewbacca, yeah. I think it's cute to put Chewbacca in there. Wookiee, right? And yes, the BK, yep. But don't tell them I said that. I don't want to get in trouble. It has nothing to do with BK. Now, prions could come from beef, but not BK beef, I'm sure. You should switch to the non-meat burgers. They're great. Impossible. I love them anyway. Yeah, so it's, I mean, look, I've been going to the bathroom for my whole life, and I'm fine. So I just wanted you to think of that, because I do. Every time I go into a bathroom, I think of it, that person who didn't flush the toilet. And yes, Patricia, they're good and bad viruses, but not because they want to be bad, because they don't want to be anything. But disease is an accident, I think, because the vast majority of viruses in us have no issue at all. This is interesting. My employer requested I repeat a series of Hep B vaccines because my antibodies were low. I think that it's different from COVID. Because there isn't antigenic variation with Hep B, and I would say that. So what's going to prevent infection is antibody, but. And you see the T cells are going to exacerbate infection. So I just don't know. That's a good question that I'd have to look into for Hep B vaccines. I don't want to just give you the wrong answer right now. Yep. Hep B persists because infected cells don't present MHC. There are some antagonists, but the real issue is that the CTLs get exhausted and stop killing, and that allows virus to regenerate. But there are evasion mechanisms for sure. You can find them on some of the slides that I've shown you. Oh, this is interesting. They're all interesting, by the way. I'm just saying. Is the asymptomatic persistent infection evolutionarily older, which have simply adapted better? I mean, that's one way to look at it. It makes perfect sense, right? That these viruses have been evolving with humans and their ancestors for many, many, many years and have reached some equilibrium where both the virus and the host have changed over the years. I think it's a wonderful hypothesis, but that we don't have any data, and I don't think we could ever address it. Now, some of the viruses that have recently come into humans, like HIV and now SARS-CoV-2, we can look at those, but it's going to take a long time to get to the point where we see some change, I think. Do I know virologists studying the human virome? I do. Yeah, we can get them on TWIV for sure. We've had one, Eric Delwart, Delwart. Search for that name. He works at a blood bank in San Francisco, and he looks in blood for viruses, which is one way to address that question. And we, Rich Condon and I actually went to San Francisco a number of years ago, went to his blood bank, and we did a TWIV there. So check that out. But Eric has continued to do very interesting virome work. And part of it is, I mean, blood is an easy tissue to get, right? Other tissues are harder, so there's much less information. And then once someone dies, it takes a while to get tissue, and then you don't know what's there or not. So, are there any stats covering historical trajectory of persistent? I don't know of any. I'm going to show you the current incidence, but I don't know. That historical one would be interesting. Does liver transplantation clear you from HBV? I can. It depends on how the viral load in you, right? Because if you have still virus in the blood, it's going to reinfect your new liver. So this is where transplantation infectious diseases come in. They have to see what you have and make sure they're treating you for CMV or Hep B or whatever. But it should, in theory, right? Arriving at the Penn Station, horrified that I need to use the bathroom. Well, I have a clue for you, all right? Go to the Moynihan, the new edition, the Moynihan thing, which is under the old post office. The bathrooms there are immaculate. Nobody uses them. Nobody knows to go there. And you're coming from, your train is going to come in there. The old Penn Station, which is under Madison Square Garden, the bathrooms are a pit because they're full of homeless men, at least in the men's room, washing themselves, but also there's a lot of urine in the toilets. Is it possible for any vaccine to spread? Yeah, if you reuse needles, you can spread other things. Now, yeah, so smallpox, you're scarifying the skin with the needle, right? And if you don't clean it and do it with the next person, boom, 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 yeah, you could definitely spread because it has blood on it for sure. Yeah. I'm going to make sure I use my mask when using public toilets. Well, right now we all are, right? I mean, when I take mass transit, I have to wear a mask, so I do. That's a good point for the future. You could just whip it out and put it on. And if someone says, hey, man, the pandemic is over, what are you wearing that for? You say, there's a lot of viruses in these urinals. And they'll say, what? And then you send them to this course. Okay, so here we have some information. Lots of formerly in Hep C infected livers treated and then used for transplant. Okay, I don't know anything about it. I'll take your word for it. Biggest life-threatening risk is to develop cancer. Yes. Well, is it more frequent in adults? It takes 20, 30 years, right? So by definition, it's going to be in an adult. And those people are most likely to have been infected as a kid, at least for Hep B, right? Because those are the ones where it's persisting. So the prion is the host protein, which is misfolding, yes, and the SSPE, it's the viral protein that's present in the genome, yeah. Guys like the urine story, huh? Yeah, they always do. Am I covering some of the viruses that present fake MHC? No, it's a viral protein that mimics MHC. We're not going to do it anymore, but I've talked about it briefly. I didn't provide any details, no. Patricia, I will check to see if anything has been changed on your account. I'll write it down here, Patricia. And maybe the mods have already addressed this. Okay. But they can't make that change, at least so far. Yeah. Sounds like you could have an entire PhD project originating in the old Penn Station restrooms. Yeah, you could. In fact, years ago, Ian Lipkin went out all over New York. Many people have done this, swabbing different things, railings, subway poles, the buttons on ATM machines. And you know what's interesting? So people have food on their hand, right? They never wash their hands after they leave, or very rarely wash their hands. And then they use an ATM machine where they touch a railing, and you can get the microbiome of that food. So you can tell different neighborhoods of New York are associated with different foods. Like downtown Wall Street, all steak, right? A lot of money. As you move further north, you get chicken and fish. Oh, it's so interesting. Very interesting. Is there any hint that long COVID is a persistent viral effect? Well, it's an effect of the original acute infection, but I don't see any data so far that says that the virus is sticking around like Ebola virus in testes and can reactivate. But it is early, right? So maybe we'll see some. I don't close my mind to it, but I don't see any evidence for it so far. It's an interesting. It's an interesting issue, yeah. The Hep C antiviral is very expensive. So that will bring up, yes, it is very expensive. And I think this is unfortunate. But we'll talk about it in the antiviral session. Okay, that's enough. We'll turn this off. We will go back to slides. We will see what we did on the... So D is the correct answer. What's in common is that virions are produced. Genomes are present but not expressed. It's certainly not in common with polyoma, HBV, and Hep C. Shared features, liver damage, Hep B and C, but not polyoma. Kidney damage, no. So its virions are produced. All right. Let's move on to latent infections. Now, this is the ones caused by herpes viruses, and they get a separate name, but they're persistent infections. And some general properties. First, gene products that promote productive replication are not made, or they are found in low concentrations. Viral gene products. The cells are poorly recognized by the immune system, and the viral genome persists intact so that we can get a new productive infection and spread it to new hosts, which of course is the driving force in evolution of viruses. The genome can be in different states depending on the particular herpes virus. It could be a non-replicating DNA in a non-dividing cell like the herpes simplex virus and varicella zoster virus, genome in neurons. It can be an autonomous self-replicating DNA in a dividing cell. Autonomous means it's reproducing separate from the chromosome. And all these herpes viruses, Epstein-Barr, cytomegalovirus, and we got a couple of other viruses, human papilloma virus, Hepatitis B virus thrown in there, Kaposi's sarcoma herpes virus, they all do that. And we have one example with human herpes virus 6, where the genome of the virus integrates in a small number of individuals, and it replicates along with the host. We'll get to that. All right, herpes simplex virus, the virus that stays with you. Unlike love, herpes is forever. Over 80% of the U.S. is seropositive, and I'm sure it's more than that because we don't do a good job of sampling everyone. We don't have to sample everyone. It's a statistical extrapolation from small samples, of course. Over 80%, and they have genomes in their peripheral nervous system. I must have, although I have never had this conversation with my wife last night. She said, "Did you never had a cold sore?" I said, "No, I never." She said, "When you were a kid?" I said, "How do I remember a kid? I don't remember that far back, but I don't remember having a cold sore. But I'm sure I have herpes simplex DNA in my peripheral nervous system." Millions of people do, and actually they don't have symptoms like me. They never get a cold sore. But 40 million people in the U.S. experience recurrent herpes disease in the form of cold sore or genital lesions. There are two herpes simplex viruses, herpes simplex 1 and herpes simplex 2. And we used to say 1 infects the oral mucosa, 2 below the belt, as my colleague Saul Silverstein used to say. He's the one who told me, unlike love, herpes are forever. Saul Silverstein was a character. He worked on herpes simplex viruses. But now we know that the two viruses can infect different. The oral can go to the genital region and vice versa because people like to mix those areas, as you know. And I think this is a well-adapted pathogen. It, for the most part, causes a mild disease and manages to be in many, many people. So here is herpes simplex, primary infection of ganglia. So when you're a kid, you get your first herpes simplex virus infection. Often you're infected in utero or during birth. The mother is seropositive, and she passes it on to you. 80% of babies get infected either in utero or during birth. Incubation period, 2 to 12 days. Virus here we're showing infecting in this case the skin, but it could be a mucosal surface in your mouth, for example. Viruses entering at the surface, reproducing, which is something that we have talked about, and virus particles are released on the basolateral side. This primary infection is usually inapparent, but can in kids have fever, sore throat, lesions, gingivostomatitis, edema, swollen lymph nodes, anorexia, malaise. So it depends. It can be mild, asymptomatic, or any of these things. So what happens then? This virus enters nerve endings. And the virus will enter the nerve ending and move to the cell body. And there the virus becomes latent. And the way the virus moves, we just talked about in a recent TWIV, grand theft kinesin. Apparently the virus particles have a motor in them that allows them to motor up to the nucleus and then to deposit, the capsid docks onto the nuclear pore. The DNA comes through the pore into the nucleus, and then it becomes latent in these. So a ganglia is a collection of nerve cell bodies. And the peripheral ganglia are outside of the spinal cord and brain. I think we have a picture of that. So we have an establishment of latency. So here's a cartoon of that. Here's your epithelial surface where you've had your first herpes infection. The virus released enters the nerve ending, travels all the way up to the cell body, and then gets into the neuron nucleus. So this is a ganglia. It's a collection of neurons. And the genome is silenced. No longer is transcribed except for one RNA, which we'll talk about in a moment. No proteins are made and just one non-coding RNA. It's coded with nucleosomes. It's silenced. Multiple copies of episomal DNA remain in the nucleus. I have a number for you. What is it? I had it from 10 to 1,000 per nucleus. And that's it. It stays there. Doesn't have to replicate because neurons do not divide. So this DNA is sitting there, latent, it's quiet, not making any proteins, and that's why it's forever. Drugs and vaccines cannot get rid of this herpes DNA. And despite the thousands and thousands of comments on this channel for the last 10 years, 12 years, who say, I've been cured of my herpes. No, you have not. It's not possible at this point. Maybe someday we could remove the genomes with CRISPR, but right now you can't. The DNA is going to be there. You may not get a cold sore, but it has nothing to do with whoever treated you. Herpes is forever because those DNAs are in your ganglia, right? The only RNA made is called the latency-associated transcript, or LAT, which is processed to make some microRNAs. There are no proteins made from this latency-associated transcript. It's a non-coding RNA. We think that the microRNAs are used in RNA silencing of certain genes to maintain the viral genome in its latent state. We're also quite clear that the compacted chromatin is important for restricting transcription, as you'll see in a moment. And clearly the host is also making some contributions as well. So that's the state between your first infection and the next. Then we have reactivation. I'm going to tell you what causes reactivation in a moment, but a small number of neurons in a ganglion reactivate. So there are most likely multiple neurons in this ganglia which contain herpes simplex DNA. They periodically begin to make viruses. Remember, the DNA is made in the nucleus. It's packaged in the particle in the nucleus. Then the capsid comes out. It's transported by anterograde back to the cell to the surface, the epithelial surface there. And you will see virions in mucosal tissue that's innervated by these ganglia, and then sometimes you get a cold sore, a blister. That's the consequence. These viruses released at the end of the nerve termini, they will infect the epithelial cells, and the virus will spread from cell to cell, and you get a blister. And that's how you transmit infection. Now, you don't need to get a blister to transmit, obviously. Many people make virus periodically without having a blister, and they transmit it really well because in general, when someone has a cold sore, you tend not to kiss them, I would think. But if you don't have a cold sore, you wouldn't know. And that's how this infection is transmitted. Now, the immune response is too slow to prevent shedding. Eventually, it will stop the infection. The CTLs will kill the infected cells. But by the time that happens, the virus has delayed it by its antagonistic proteins that we've talked about before. You do shed enough to infect someone else, and that's all that matters. It's kind of like humans, you know. If you have a kid by your 30s, what happens beyond that doesn't matter. You've ensured the persistence of the species. We're evolved to have kids early, and then that doesn't matter, those late diseases. The same thing with virus here. You can spread to a new host, and then it doesn't matter if the immune system knocks you out because your genome is still in the peripheral ganglia. Some people reactivate every 2 to 3 weeks like clockwork. Some people never do. Some people do with blisters. Some people do without blisters. And that's where type 1 goes, the trigeminal ganglia. It says about 20,000 neurons here. It's a collection of neuron cell bodies outside of the of the central nervous system. Obviously, it's outside of the brain here. And you can see all the nerves coming from the ganglia, innervating your lip, your nose. You can get cold sores at any of these places because the virus can go in, stay in the ganglia, and then come out any of these places. And sometimes, as I said before, the virus goes the wrong way into the CNS. This is connected to the central nervous system, and then you get encephalitis, which is a problem. But most of the time, it doesn't do that. And so, again, 10 to 1,000 genomes within these ganglia, and multiple cells are infected. So if you have genital herpes, then obviously there are ganglia down there that are going to harbor the virus as well. They're just not called the trigeminal ganglia. You know how we found out that this is a place? So you may know of a disease called Trigeminal Neuralgia, which many people develop. Terrible, terrible pain in this region of the face. And one of the treatments is to remove the trigeminal ganglion. And that may or may not treat it, but they did some studies and found that that's where herpes genomes were. Be hard to do otherwise, right? So what reactivates? Sunburn, physical or emotional stress, nerve damage, hormonal imbalances, steroids, a lot of different things. Some people go skiing, the high altitudes with a lot of UV, boom, they get a cold sore. What happens? This is such a great story. These things stimulate the production of viral proteins to activate the transcription program. Now, remember, you have to remember back from transcription. When the genome comes into a cell, the particle itself actually delivers a protein called VP16, which is a transcriptional activator of the immediate early promoter. This promoter is not recognized very well by cellular machinery, so the virus needs to bring in this protein. But in a latent state, there are no viral proteins. It's just the genome. So how in heaven's name can this get reactivated? Aha! I'm going to tell you right here. So here we have the ganglia. We have a neuron in there where we have latent genomes. They're sitting there, they're just making lat transcripts, but they're not making anything else. So there's no VP16. It turns out that an important component of the silencing of this genome is methylation of histones. These are ME stands for methyl, and these numbers refer to different histone proteins and different amino acids. But essentially, the promoters are all silenced because the DNA is wrapped around histones, these little balls here. The DNA is wrapped around histones, and it's tight because of the methylation. So remember, when foreign DNA comes in the nucleus, it's chromatinized, it's wrapped around nucleosomes, and it's methylated to shut it down so it can't be transcribed as a defense against invading DNA. So that's the state of herpes DNA. So how do we reverse it? Well, when you have neuronal stress of some kind, you're taking an exam, you get stressed, turns out that activates protein kinases in your neurons. There are stress-related protein kinases, and one of the things they do is to phosphorylate these histones. And so here we have these little blue ovals, PS10. So this is a phosphorylation on serine 10, which happens to be on histone 3, right next to a site of where the histone is methylated. It turns out that phosphorylation provides a temporary reversal of the silencing of this genome. And so for a brief time, you get all classes of the mRNAs made, immediate, early, and late. Normally, you wouldn't have that. You would have first immediate early, then you would have early, then you would have late. But here, when this phosphorylation occurs, you get all of the promoters active. And why is that so important? Because VP16 is a late gene, is encoded by a late gene. I just love this. So VP16 is made, and then the virus can go through its transcriptional program. It encodes, it stimulates the demethylation of these histones. It has ability to do that. And then you get full reactivation. So the methyls are gone and the DNA's the histones acetylated so that you can open up the chromatin and boom, reactivation. So that's how it happens, all through stress-related kinases. Oh, it's just amazing, isn't it? I just love it. Okay, persistence of herpes simplex virus in nerve ganglia requires which of the following? A, continuous episomal DNA replication. B, low-level production of virus particles. C, silencing of all gene expression except LAT and microRNA. D, UV light stress or steroids, or all of the above. And while you're chewing on that, let's go to the next question. Do fans of TWIV have some group name? I don't know. I think the people in the chat could answer that. TWIV fans. There should be something better, right? Saw someone with a lesion on her thigh, which she said was herpes. And I'm not aware of it on the thigh, but certainly varicella zoster could do that. But I don't know. What would happen if RNA silencing did not work? I think you wouldn't get establishment of latency, most likely. I think, I'm not sure if the neuron would be killed or not. That's a question I've asked many herpes virologists. You know, when you get reactivation, is the neuron killed? I'm not sure it is. So you would not get latency, but the consequence for the ganglia is not clear. Does every persistent infection in humans have a homologue in chimps? I would hate to say every, but chimps have some of our viruses, but not all of them. They may have related viruses. Because remember, we have a common ancestor. We didn't come from chimps. We have an ancestor in common. Sir, why does our immune system not make memory antibodies against HSV and prevent the blister? It does. We do make antibodies, but remember, the memory response takes a couple of days. And in that time, this virus has made particles already, and they're out there. So it's just enough time to transmit, and that's it. Then the antibodies could come, and the T cells could come. That's fine, but not a problem. I see the quiz is not active. Sorry about that. Okay, thank you. I guess the texting worked. Okay, the quiz is now activated. Thank you. Sorry about that. Got all these texts all of a sudden. They all just came in at once. There's a little bit of a delay. Thank you. Good. Do I think vacuum toilets are better than standard swirling? I would think because they're pulling down, right? But I'm sure there's still some aerosol generation. But the one thing is that people can't leave urine in them, right? Because there's no liquid, I think, if I remember. Yeah. But of course, yeah, if you put the lid down, that would help. But in the urinal, there's no lid, so that's the problem I have. And people of my sex, right? Is there any advantage for the host to reactivate? No, so the host, the neuronal kinases that get turned on by stress, they have other functions related to host survival. So the virus has just evolved over years to tap into that to be reactivated. Yeah. Okay, now we have quiz questions. Thousands. So the thing is, it did take like 3-4 minutes for me to get your texts. So it's not immediate, but it's better than nothing. Twivsters. Twivsters is pretty cool. I like that. Can we define HERVs, human endogenous retroviruses, as a category of persistent infections? I think so, yeah. The thing with humans is that they're not making infectious viruses, right? But they're certainly latent. The genome is there. So I don't know if it's persistent when the genome doesn't work anymore. But for a time they were, yeah. I don't see why not. Is herpes reactivation the cause of Bell's palsy? I'm not aware of that, no. Not aware of it. A lot of good names here. Let's see what we're doing with the quiz. 42 out of 94. I'll give you a couple of minutes to yeah, so the texts were sent quite 8 minutes ago or so. It's okay. As long as I got it. I saw it here. Yeah, I got 3 separate texts from my 3 mods who are here today. Thank you very much. But there is a delay, so don't worry about it. So we got 46 out of 94. Anybody else taking? A lot of good names for Twiv fans, yeah. Is it possible to competitively block the phosphorylation site? Yeah, but the problem is you'd have to get the drug into the neuron, which is tough, and you'd have to not have host effects, right? So I think that would be a challenge. Is there any link between reactivation of latent viruses with neurodegenerative diseases? There are. Many people feel that there's a strong link between herpes infections and Alzheimer's disease. And there's quite a literature on this. We've done a TWIV on it, but the idea is that every time you have a herpes reactivation, some virus gets in the brain, and the brain responds by making proteins that are supposed to be defensive, but which may lead to Alzheimer's. So that's being tested in clinical trials right now. How is it possible that there's the same? So there are signals on the histones that are known to be methylation sites. Not every histone amino acid can be a methylation site. And there's a language of histone sequences that specify methylation, phosphorylation, and so forth. Yep. Can shingles vaccine prevent or reduce herpes simplex? No. Very different. Very different. Is herpes linked to trigeminal neuralgia? No, it is not. Okay, let's move onwards now. What's the result here? Let's bring up. Persistence requires which? The answer is silencing of all genes except LAT and microRNA. Continuous episomal DNA replication. No, there's no DNA replication in the neuron. Low-level production of virions. No. Remember, nothing is made. UV light stress or steroids is needed for reactivation, not for persistence. so it's not all of the above. Next one, Epstein-Barr virus. Again, most adults seropositive carry the genome. This is another herpes virus that stays with you forever. The genome resides in B lymphocytes. Most people are infected at an early age and are asymptomatic. Although it's very interesting, a number of studies have been done which show that if you weren't infection at a young age, you get infected in college, typically. You start kissing people, and then you get transmitted. And interestingly, the studies have shown that people who do not kiss other people, meaning not their family, don't get seropositive. Really interesting. So this is an agent of infectious mononucleosis and some human cancers that are quite important, as you can see here. So the primary infection arises at a mucosal epithelial site, very much like herpes simplex, typically transmitted by saliva given to you by a kiss from either your parent or a loved one. The virus infects the epithelial cells. It is released on the basolateral domain and infects resting B cells, of which there are B cells underneath here, right? Because that's their job, to go there and make antibodies for other things or to patrol looking for antigen. And the virus actually can transform these B cells into memory B cells. A very few of them become latently infected resting memory B cells, and they are essential for latency. These viruses can reside in many areas. They can reside in lymph nodes and so forth. Eventually, many of the B cells are killed by infection, but a small fraction are transformed into resting memory B cells. And periodically, these will become reactivated. Many of them can be killed by the cytotoxic T cell response, but a fraction will escape killing the B cells. Here we're looking at the blood vessel down here. The B cells will go into the subepithelial tissues. They're making infectious virus, which then infects the basolateral domain of the epithelium, and then you shed virus in saliva to infect others. So the B cell is the essential site of latency. Here is a virus that can transform B cells, and these are memory B cells, so they're long-lived. And in that, it stays with you forever. So very different from herpes simplex, right? There's no infection of ganglia, but in this case, it's memory B cells. So the DNA is a self-replicating episome. It's nucleosomed in B cells, so it's silenced. Produces more gene transcripts than we saw with herpes simplex virus, but certainly limited compared to the full lytic cycle. The B cells, as I said, they go to the bone marrow and lymphoid organs where they stay forever, essentially. And they're not killed unless reactivation occurs, and that would put viral peptides on the cell surface. But there's a lot of modulatory proteins encoded by the virus which delay CTL killing enough so that the B cell can get to the epithelial surface and to below the epithelial surface and release virus for transmitting to others. Our next one is varicella zoster virus. Chickenpox is the varicella part, and shingles is the zoster part. So you typically get this as a kid. This is transmitted by respiratory droplets now, and it will infect your upper respiratory tract, reproduce in primary lymph nodes. You get a viremia, which moves the virus to the liver, spleen, and other organs where it reproduces more, and then you get a secondary viremia. This should sound familiar. We've talked about this as a classic means of spread. And that secondary viremia, after about 14 days, brings virus to the skin, and that's where you get that first rash of chickenpox. So that is quite a painful rash, and it is over the entire body. So it's something that we'd like to prevent by vaccination. But the problem is then the virus goes latent in ganglia, neurons within ganglia that innervate the skin. So the virus enters the nerve endings and goes to the ganglia. They become latent, and again, they are not silenced to the extent that herpes simplex virus is silenced, but a restricted repertoire of gene products are made there. And then many years later, they can be reactivated and virus will move down to the skin and you get the typical rash, which is in typically an area called a dermatome, which is innervated by a single set of nerves. So here is the typical rash on the trunk, the abdomen, along the ribs. So that's shingles. And so we have many people who had chickenpox as a kid still, and they're at risk for getting shingles. So we vaccinate them against shingles. There's a Shingrix shingles vaccine, which will prevent zoster. And of course, for kids who have been vaccinated, they shouldn't be developing zoster, although perhaps a fraction of them will because the vaccine is an attenuated vaccine. So before the vaccine, 99% of adults would be infected in childhood. 30% would develop zoster, typically over 50 years later, most of them, but not all. I know several young people who were on immunosuppressive therapy who developed zoster. Latently is comprised of episomal DNA, 2 to 9 genomes and 1 to 7% of neurons, non-replicating genomes. And as I said, the gene expression is restricted. What triggers reactivation, we really don't know. For herpes simplex, beautiful work showing the phosphorylation of histones, but here we have no idea what is triggering reactivation and why it happens so late in life. Cytomegalovirus is another herpes virus with high seroprevalence, transmitted by respiratory routes. The virus is in the saliva. It can be found in urine. It's transmitted by sex as well. Replicates in peripheral blood leukocytes and endothelial cells. And here's a graph of seroprevalence in terms of years, age in years, in different racial populations here. And you can see that seroprevalence rises approaching 100%, 90 or 100%. The primary infection usually gives you an asymptomatic or a febrile mono-like illness, tired as for Epstein-Barr virus. You can shed virus in saliva and urine for months to years, so infectious virus. Eventually, this will be resolved by immune responses, but you do have latently infected myeloid cells, right? These are cells derived, the common myeloid progenitors, for example, which are precursors of monocytes, macrophages, and dendritic cells. And so the virus persists in that form. This is a big deal in organ transplantation because everybody has this virus, and when you're immunosuppressed so you don't reject the organ, it can reactivate cytomegalovirus with pathogenic consequences. The other issue is that this is one of the torch agents that can cross the placenta. So a pregnant mother can transmit the virus to the developing fetus, and that baby can have multi-organ congenital defects and deaths. And so this is a chart from the CDC which shows you the numbers. So 1,000 pregnancies leading to live birth. If 600 of those women will have CMV before pregnancy, most of them give birth to negative babies, but a few do have CMV positive babies, and 1 or 2 of those will have permanent problems. So 1 to 2 out of 1,000 live births. 400 of these women do not have CMV before pregnancy. Most of them don't get it during pregnancy. Some do. 5 out of those 7, 5 give birth to CMV negative, 2 positive, and of those, the 1 will have a problem. So you can get an idea. The frequency is quite high because this is 1,000 and many, many people are giving birth. So you should try to avoid becoming infected if you're not already infected. And we don't have a vaccine for this. So we do have antivirals, but this can be a real issue. All right, let's turn on the next quiz this time. 4, very good. What do persistent infections with EBV, VZV, and CMV have in common? B cells are essential for latent infection. Many cause congenital birth defects, viral DNA persists as an episome. The factors governing reactivation are well known, or all of the above. Okay, let's do some questions here. This is an interesting approach, or an interesting interpretation. Viruses sound less and less like a wrench thrown in a machine and more like a slight fluctuation in voltage nudging a poorly maintained machine to have the breakdown. I don't think they're a wrench in the machine. I think it's quite a subtle thing. Is herpes going to replicate better in brain cancers? I don't know. I'm not aware of any data that would suggest that. How does Shingrix work? It doesn't keep virus from reactivating, but it neutralizes viruses that are produced rather quickly. Do viruses talk to each other when an individual is infected? I don't know. That's a good question. There's some evidence in bacteriophages that infected cells can communicate via peptides. So I think the possibility is open that other virus-infected cells would do that as well. How does valacyclovir work? Well, what it does is you get a cold sore, and then you treat it, and it stops virus reproduction, so the sore goes away more rapidly. It doesn't last as long because it's a really good inhibitory drug, as we'll see later. Yes. So is there a molecule through which viruses? So as I said, in bacteriophage, maybe, but nobody's reported that in eukaryotic viruses. But why not? Thank you, John, for your contribution. Glad you liked it. I had chickenpox. Yeah, so did I. Yeah, I had chickenpox because when I was a kid, there was no vaccine. EBV can infect memory, which are produced for, just memory B cells, any memory B cells, as far as I know. But I don't think there's a negative consequence to that. Odd pediatrician here. I used to see shingles regularly in my practice. Now I only see it in the unvaccinated. So you think that's because of Shingrix, do you? That's interesting. Oh, so you saw it. No, but you're a pediatrician, so you used to see it in kids, huh? And why don't you anymore? Because of the vaccine, I presume so, right? Mono-like illness is something that EBV causes, which is fatigue, right? Mononucleosis. Yes, so Mike is saying shingles in kids, right? Interesting. What was it? What would be the frequency of that? Is there a way to test whether one has VZV latent? No, you can't really, because you'd have to take out ganglia to look for it. I think you just have to assume it's there. Yeah. All right, let's go back to our quiz. So the answer is the DNA persists as an episome. That's in common between EBV, VZV, and CMV. B cells are essential only for EBV. Birth defects is only for CMV. Reactivation is not known for any of these, and so it's not all of the above either. Okay. Last herpes viruses, number 6 and 7. These cause rashes. Exanthem subitum, our pediatrician here should know about this. Called sixth disease, right? After all those 6 childhood rash diseases, Most kids get infected. 85% of adults have antibody to both viruses. Respiratory secretions, parent to child, is the way they're transmitted. These viruses infect a variety of cells, and they become latent, well, for HHV6 in monocytes and macrophages and progenitors of stem cells, essentially, and for 7 CD4 positive lymphocytes. So different cells in both cases are the sites of latency. But what I want to tell you about, and these are rather mild infections, what I want to tell you about is that, at least for HHV6, in some cell types, the DNA integrates into telomeres. The telomere are the ends of your chromosomes, which we'll talk a bit about on Wednesday, actually. And the genome actually has telomeric repeats at its ends. There's the HHV6 genome with direct repeats on either end. It's got a little bit of a telomere there, which allows it to integrate into our host chromosomal DNA. About 1% of transmission it involves integration, and those people will pass it on to their offspring. And the virus can be reactivated when those kids are born and grow up and shed. So it's quite interesting. It's very rare. It's only about 1%. But it's clearly there, and it's, of course, a good strategy for latency and transmission. The telomere is a conserved end, so maybe it's a good place to integrate anyway. The 7 doesn't do this as far as we know. So let's end up with this slide, which is a estimated burden of chronic viral infection in humans. And let's make this big so that we can see it. So this is estimated from the percentage of humans carrying a given virus. So we have estimated number of infected individuals, and then we have different viruses here. And here we have everyone, because everyone has an endogenous retrovirus, right? And this is assuming the population is slightly less than 7 billion. This was done a number of years ago, 6.75 billion. And so here, this is everybody up here, because everyone has ERVs. And remember, this is based on seroprevalence data, which mostly is done in developed countries, so it's an underestimate of the global seroprevalence for some of these viruses. But you can see there are many, many billions of chronic infections. We have Anelloviruses. These are little single-stranded DNA viruses. Pretty much everyone has all the time. These are chronic viral infections. Then we talked about HHV6 and 7, VZV, EBV, CMV. Then we have our polyoma viruses. So this is only slightly going down because of underestimation, I think. Herpes, even adeno-associated virus is quite high. HSV2, not quite as high, sexually transmitted. Then hepatitis B virus, papillomas, Hep C, HIV down here. This is like 35 million people. So we each have about 8 to 12 chronic infections, at least, maybe more. And most of us don't show any symptoms of these infections. So I would say they're part of our metagenome. They're part of us. Viruses are us, folks. They're not just the enemy. They are us. And I think the key here is that we need to consider what these infections mean for humans without overt disease, and the need to understand chronic diseases that are considered, quote, non-viral, end quote, based on our inability to compare infected to non-infected individuals. All right, just think of that. You have a disease you're studying, and you're comparing people with and without disease, but they're both infected. So how can you dissociate these chronic infections from chronic diseases? Whatever chronic disease, whether it's cancer, diabetes, or hypertension, or heart disease, you do not have a controlled population that lacks these virus infections. So you can never say that they have no role. I think it's really important to remember that. Very few do. And that's what I want to leave you with for today. Next time, Wednesday, we are going to talk about transformation and oncogenesis. About 20% of human cancers are associated with viruses, and we're going to talk about how that works. Viruses are us, folks. Viruses are us. Let's see what we have here. Any treatments for mono-like illness? Not that I am aware of. Can I please make a lecture on hepatitis? So I'm going to teach a course on viruses next. So this one was called Virology Live. The next will be Viruses Live. And we'll talk about all these viruses. But it's going to be a while. It's probably next summer because I have another course to teach in the spring. And I talk about how it integrates. I don't really know much. I don't think much is known about how. All we know is that the target is the telomere. By homology, the virus genome has homology with the human telomere, and that probably drives integration by recombination, but we don't know much about that. Is there no vaccine for herpes virus? No, there is no vaccine. There are some in development, but another thorny one to make. Chickenpox, respiratory, it's on that slide, respiratory droplets. It could be, saliva, but most likely kid-to-kid respiratory droplets. Shedding before the rash. Rabies virus is useful for mapping neural circuits. Yes, so is herpes virus, the pseudorabies virus, for example. Haven't really talked much except in the lecture on pathogenesis. We talked about using herpes viruses to trace neural circuitry, but not much more than that. Does human herpes 6 have any influence on aging? Not that I know of, because it integrates into the telomere. No, I don't believe it. I mean, you're thinking that it keeps the ends of the DNA from being chewed back. Yeah. Listen, no, those people have the same longevity as others, I believe. Could measles virus be used as a treatment for B memory cell infected with herpes? I don't think you should use measles as a treatment because it's going to give you immune amnesia. So not a good plan, I think. How is an episome defined? Episome simply means a piece of DNA that's not integrated into the chromosomal DNA. It's just on its own. It could be a circle. It could be a linear piece of DNA, just separate from the chromosome. While you're here, folks, 218, that's pretty good. If you haven't liked us, give us a thumbs up down there below the screen. Does the HSV in the nucleus have any specific locations? No, they're floating around. They're episomal. Remember, not integrated. Perhaps the biggest barrier I find to truly understand everything you have to say, everything, I'm sorry, is my own lack of knowledge about genetics. Do you have a video or course on intro? I don't, but there are many others out there. But the question is, which one is going to help you the most? But, MIT has wonderful courses. I would look into that. I will actually search for something that might help you. Does high AAV seropositivity limit application for gene therapy? Yes, but you can use different serotypes, or you can modify the serotypes, actually. And we did some papers on TWIV where they modify and make a brand new serotype that no one has seen before. So that's how you can actually get around it by doing that. Really cool. if two different viruses were able to infect the same cell, it's like two viruses walk into a bar, would they have the ability to suppress each other? Yeah, they could. So I did an experiment years ago with a colleague of mine who works on adenovirus. We co-infected cells with adenovirus and poliovirus. And they can have a one-way inhibition. Usually one predominates. Polio did because it reproduces faster than adenovirus and shut off translation. Thank you, Pam, for your contribution. Really appreciate it. But of course, as Jeff said, you could also have synergism as well, for sure. Not been studied very much. HSV2 is not integrated. It is episomal. Yeah. Thank you, Barb Mack, for your contribution. And yes, we bring you into the viral world. And we will talk about the SARS-CoV-2 variant tomorrow on TWIV. We will talk about it on Wednesday evening at 8 p.m. Eastern on Q&A with A and V. And I think, could an outbreak of measles put us back to square one? That would have to be quite an outbreak, right? I would think you would have local issues. So if a community had a measles outbreak, then yes, they would have to be revaccinated. I doubt it would happen globally. Yeah. Here we go for a lectures on genetics. There you go on YouTube. That's a good idea. Are there studies of persistent infections in CFSME? Yes. Yes, there are, but nothing consistent turns up. That's the problem. Oh, this is good. Two viruses walk into an Epstein-Barr. Oh, very good, Thor. Well, George, viruses are an organism with two phases. There's the inactive, non-alive particle and the live infected cell. Go back to lecture 1 and I talk about that. But they are pieces of information, but then so are we, right? Thank you for your contribution, Kate. And Nels is trying to schedule a TWEVO. Nels is tough with, it's his job to schedule the TWEVOs, and he's a busy guy, but I hope to have one in December. Yep. Yeah, there's a Prion lecture later on in this course. Yep. It's called Unusual Infectious Agents. It's pretty much fun. Okay, I think that'll do it for today, folks. We'll be back Wednesday, talk about persistent infections. Until then, thanks for coming. Thank you to the mods for helping out today. Really appreciate it. And everyone be safe and think about viruses. Bye-bye.